So my name's Mike Fay. I'm a radiation oncologist. I was previously a medical oncologist till I got sick of making people sick all the time and changed to radiation. So I know a little bit about what the previous two speakers have talked about, but I wanted to talk a bit more about imaging and what we're doing at HMRI. So a little bit about glioblastoma, which is the sort of tumour that I spend most of my time working on. It's pretty uncommon. It's about 1% of the cancer burden. But the big problem, I think, is that in terms of lost productivity, it's absolutely huge. Often patients are young, 50% of them are under 65, and they estimate that the hospital costs alone are $50 million a year just from this disease. We've got a long way to go in terms of treatment outcomes. We think that radiation is a pretty effective modality, but I think there are better ways that we can use it, and I wanted to show you some of these things today. And unfortunately, recurrence is fairly common, but I think the good thing in this disease is that even small incremental improvements can have a huge impact. We had a look in Queensland a few years ago. I moved down from Brisbane about 4 years ago, and we looked at over 2,500 patients with GBM, and we looked at the survival over 20 years. And it clearly shows us that there's a long way to go. The survival at 24 months was 10%. When we looked over the last 10 years, that's significantly improved. So it had gone up to about 18%, but it's still, there's a long way to go with this disease. So I was going to talk a little bit about the problem of GBM, talk a bit about brain tumour imaging and some of the MR imaging and PET imaging research that we've been doing, and then show you some of the basic science stuff that we're doing at HMRI. And then I want to talk a little bit about a new emerging area called theranostics. So we were visiting a zoo in Germany when I was doing a sabbatical there a few years ago, and we saw these monkeys, and I sort of thought this is a little bit like the problem we've got in glioblastoma. Something's holding us back, but it's kind of hard to work out what it is. Pretty easy to work out what's holding this fellow up. But so I wanted to talk a little bit about the tumour. So this is a picture of a brain tumour, and you can see the area here that's involved with the tumour, and it's slowly invading into the rest of the brain. And until Craig Getty stole my cane toad analogy, what we used to say in Queensland was that there was this sort of invading front of cane toads that are sort of the stem cells for the tumour that are spreading into the brain. So we've gone then from looking at the gross pathology to microscopic pathology, and nowadays everything's moving towards genetics, and it becomes completely unintelligible, but there's a whole lot of genes that can be upregulated, and so we're moving to think more and more about how we can look at the gene mutations and target them. So when I started training in radiation 20 years ago, this was how we would plan the treatment. And I remember one of the consultants saying to me, well, you try and find the cavity and you focus the middle of the field in there, and you're probably likely to hit the right place. That was a little bit scary, and thankfully things have changed rapidly. So then we moved to CT, and you can see exactly where the craniotomies happened. You can see the deficits there, but you really can't see where the tumour is. And then MRI came along, and we got a much better view of where things were. Except it's a little bit too light, sorry. But the trouble with MRI is that there are multiple different MRIs, and some of them look like the tumour's more extensive than it is in other sequences. So we started looking at PET scanning, and PET uses traces that go into the brain tumour and are taken up by the tumour, and hopefully are not taken up by the rest of the brain. And this is one of these scanners, and we've got two of these in Newcastle at the moment, and we're starting to use them more and more in terms of finding out how we're doing with these sort of tumours. So I wanted to show you some work that we did in Brisbane in terms of where the tumour was. So this is an MRI scan. You can see the white area down the bottom, and you would think that that was mostly where the tumour was. But when we look with PET scanning, we found that actually the area that the tumour was involved with the tumour is mostly above that area. So this area up here, it looked a little bit abnormal, but this looked the most abnormal area, and that's actually looking quite good on PET scan. So sometimes the information that you're getting to tell you where the tumour is isn't as accurate as we would want. Here's another example of a patient where it was pretty subtle that there was something sort of abnormal here, but then when you look on PET scanning, you can see an awful lot more clearly where the tumour is. So this is helping us work out where the tumour is. At the same time, the radiation technologies are developing rapidly. You'll notice that radiation oncologists don't wear ties, and that's because we don't want to spread infection, but also we don't want to get them caught up in our machinery. So that's another advantage of radiation. But with this new technology, what we're trying to do is to shape the radiation beam. So in the old days, we might have treated with 2, 3, or 5 beams, perhaps. With this new technology, we can treat with 10,000 beams as it travels around the patient, and we can really shape the radiation treatment much more tightly to where we want to go. And that means we can avoid normal tissue. We can try and stay away from the normal side of the brain, just treat the side that's involved. But we really need to know where the tumour is, because the more conformal, the more tightly you keep your radiation field, the more you want to know exactly where the tumour is. All right, so the other thing that's been really tricky in radiation, and indeed in brain tumours, has been working out whether the treatment's worked. And so Craig told you a lot about large clinical trials where you're needing large numbers of patients. One thing that we're hopeful will work is that we can use PET scanning to give us an earlier idea of whether the treatment's working, and also potentially do the same thing with less patients. So this is an example of a patient where we did a scan called an F-DOPA scan. An F-DOPA is taken up by the brain tumour, but it's not taken up by the normal tissue. And sometimes they call the nuclear medicine the Department of Unclear Medicine, because everything's always a little bit fuzzy. But you can see that there's a tumour lighting up here. So we started doing this as a way of looking at how the tumour's responding, and we found some really interesting information that even though the MRI might not have changed very much, sometimes the PET scan's really looking an awful lot different. So we've been banging on about this for nearly a decade, and the government, to their credit, has finally listened, and they've just given a group in Melbourne about $1.5 million to do a national study looking at brain tumour imaging, and we're getting that study going at the moment. It's still a little way off, but I'm really hopeful in the next few months that we'll have a study going. So the problem with glioblastoma is sufficiently complicated that we need to attack it at different levels. And one of the great things about having HMRI is that we can do some basic science research. And that means doing things like growing cells in tissue culture. And if my slide will change, which it won't. Oh, it's changed now, sorry. So this is the traditional way of growing cancer cells. You have a cell line that keeps growing and growing and growing. It's what they call immortalised. And we take these cancer cells, we grow them in colonies, and this experiment that I'm showing you with 4 different plates would take about 3 weeks to do. So each plate would be a different radiation dose or a different combination of drugs to try and look at, in cell culture, which ones would work. Now, we're basically impatient and want to get moving, and doing this sort of stuff is really, really slow, especially if you're going to look at a whole lot of different radiation doses or a whole lot of different drugs. So we're really fortunate to be given a really neat bit of equipment, which is a mobile irradiator. And we had this crazy idea that if we could put a cell plate on top of this irradiator, then we could take it into the lab and look at a whole lot of different radiation doses all at the same time. So I went off to my shed and we built this thing, not that bit, just the rest of it, and that meant that we could change the height and carefully arrange the radiation doses. And we did a study where we looked at these plates and we put lots and lots of cells onto each plate. We had 96 wells, so we could get 96 different data points. And this is looking at no radiation treatment, the green ones, and we found that 3 days later when we looked at them, surprise, surprise, there wasn't much difference. Then we did it on a standard linear accelerator, a big radiation treatment machine, and then we used our little irradiator, and we got some really interesting results. We got a standard decrease in the radiation, in the cell growth in the ones that were treated on standard linear accelerator, but then we got a whole lot of different data points when we did it on our machine. And the neat thing about this is that in one experiment, we can get 96 data points. So we did it with 3 different cell lines. Some cell lines are more resistant to radiation than others. These are the more sensitive ones here. These are more resistant, what's called U87 cells. And suddenly we could get a whole lot of data. And this is just done on 3 different plates. The experiment took 3 days, and we get 96 data points for each replicate of the experiment. So we're really excited about this because it's a way of really rapidly looking at a whole lot of things all at the same time. And we were given the bit of equipment from a hospital in Melbourne for $1. So for a machine that probably would have cost us $500,000 or $600,000, we think this is going to be really useful. So just looking a little bit more of some of the things in the lab, and I don't want to get too caught up in the detail. DNA is organised in chromosomes, and then it's got these things that look like Oreo cookies that kind of wrap around the DNA. And these are the histones, the chemicals that keep the DNA all packed up. But what we can look at now is when we irradiate a cell, we can look at the damage that happens. And it looks a little bit like this. We get a whole lot of these little green spots in the nucleus, and this is where the radiations cause damage. And we know that if this damage is still there at 24 hours, then that cancer cell is probably doomed. So what we're going to do next is use some of our technology that we've developed with the irradiator and then look at some new drugs that we've got available that can start interfering with this and pushing the cell towards dying rather than being able to repair itself. Because we think that part of the problem with glioblastoma is that these cells are really good at repairing the damage that we do. Okay, so there's another really cool aspect of this, and I pinched this graphic from a company called Telix Pharmaceuticals. So the good thing about radiation oncology is that you hardly ever have to declare conflicts because the drug companies are not interested in radiation oncologists at all. But we have done a little bit of work with Telix Pharmaceuticals, also with Canna Pacific, and I'm involved with Genesis Cancer Care and with Theranostics Australia to do with this sort of work. So these are new sort of drugs that are starting to become available. And basically it involves 3 bits. So we've got a horrible looking hairy cancer cell here. Then we've got some sort of key that lets us lock together a cancer fighting drug onto the cancer cell. Then we've got some sort of fancy linkage chemistry, and then we have a payload, which is a bit like a smart bomb. And so we're using radiation, but this time we're using radiation given by injection. So this is the sort of thing shown as a diagram. We've got something like an antibody, and that'll bind to a certain part of the cancer. Then we have some linkage chemistry, and then a cage on the back of it. Now these cages are pretty neat. They can lock into a radiation molecule, and then the whole lot locks together, travels to the cancer, and hopefully causes a lot of damage to the cancer cell. Why would we do this instead of giving the radiation from outside? Well, it means that we can get a much higher radiation dose into the area that we need to, and it means we can potentially cause a lot less collateral damage. So the challenge is really to find things that are sticking out on the cancer cell, like this, sticking through the cell membrane, and they're available, for the drugs to bind to. So we got pretty excited when one of the guys that I worked with in Germany presented a paper showing that one of the standard PET tracers, a thing called PSMA, could also light up in brain tumours. And the exciting thing was that this was readily available, that PSMA is available and used for prostate cancer imaging. And the other thing is that as well as having an imaging molecule, we've also got a very high energy radiation source, a thing called lutate, which comes from Lucas Heights in Sydney. And we can put those together and potentially use that for treating the cancer. So it's been used a lot in prostate cancer. It's becoming more and more popular. But this was the picture from brain cancer. So here's a patient's MRI, and then the same thing using a PSMA PET scan. And I think the holy grail of cancer treatment is when you can move towards knowing whether the patient's going to respond. So we're really hopeful that with this sort of thing, if you can see that there's a lot of uptake, it takes away some of the guesswork in terms of is the treatment going to get to the right place. So currently we've got a clinical trial that's just going through ethics and hopefully should be ready shortly, looking at the first stage of this, which is in a group of patients with glioblastoma, how many of them show this sort of uptake and how many of them show no uptake at all, and also is there any uptake in the rest of the body, because we don't want to cause a whole lot of problems with eyesight and these sort of things. So far it's all looking hopeful. But the other thing is that this is a really interesting way of treating cancers. And I think over the next 5 or 10 years, we'll see lots and lots more of these sort of treatments being developed. So I wanted to say a few thank yous. I've worked with a big team across the country and in Germany, and I'm appreciative of all of their help, particularly the guys locally have been absolutely fantastic. And especially the Mark Hughes Foundation, who've been incredibly supportive and have really helped us get an awful long way in a short time. Thanks very much.